Welcome back to Kinesthetic Knowledge. I'm your host, Isha, and in today's episode, we will dive into a one-on-one session on muscles, particularly looking at strength and stability. Whether you're an athlete striving for peak performance or looking to improve daily activities, or simply just interested in understanding the mechanics of the human body, this episode is for you. Muscle strength and stability are fundamental to our ability to move, perform tasks, and maintain posture. Strong and stable muscles not only enhance athletic performance, but also play a crucial role in preventing injuries and maintaining overall health. In this video, we will delve deep into the intricate world of muscles, exploring foundational topics such as muscle structure and function and the neuromuscular junction, and the fascinating process of motor unit recruitment. These topics will be discussed in greater detail in future videos, so let's get started. At the cellular level, muscle contraction is facilitated by myofibrils, which are cylindrical bundles of protein filaments arranged in highly organized striated patterns. These filaments are key players in the contractile process. Actin, the thin filament, is a polymerized protein that serves as the substrate for myosin, the thick filament. Myosin, as I just said, the thick filament, is a molecular motor that utilizes ATP to generate force. The magic of muscle contraction unfolds within the sarcomere, which essentially refers to the arrangement of the thin and thick filaments within repeating units and the fundamental contractile unit of a myofibril. Each sarcomere is composed of a precisely arranged array of actin and myosin filaments. The sliding filament theory explains the remarkable process by which these filaments interact to generate force. When a nerve impulse triggers the release of calcium ions within the muscle cell, a series of conformational changes occurs in the myosin molecule. This allows the myosin head group to bind to specific sites on the actin filament and undergo what's known as a power stroke. This is essentially a ratchet-like movement that pulls the actin filament towards the center of the sarcomere. This coordinated movement of myosin heads along multiple actin filaments leads to the shortening of the sarcomere, ultimately resulting in a forceful contraction of the entire muscle fiber. So, when it comes to muscle strength, it's actually been reported that muscle cross-sectional area is a major predictor of force production. Simply put, a greater number of contractile units suggests that there are more myofibrils and sarcomeres packed into the muscle. Since each sarcomere can generate force, having more of them allows the muscle to generate more total force. However, this is not the full story. There are other factors that influence strength. Skeletal muscle fibers can be categorized based on their physiological properties as well. Current muscle fibers are divided into two types, type 1 and type 2. Type 1 fibers are known as the slow twitch fibers, which are fatigue resistant and well suited for sustained activity. Type 2 fibers, on the other hand, are categorized as fast twitch fibers and can be further subdivided into subtypes, including type 2A and type 2X fibers. Type 2A fibers exhibit a balance between strength and endurance capabilities, while type 2X fibers are best suited for brief bursts of high power activities due to their rapid contraction speed, but also fatigability. What's interesting is that many recent studies have correlated different functional capacities of the intact human skeletal muscle, and that includes endurance, contraction velocity, and strength, to the distribution of different fiber types in the muscle. A number of these studies have actually indicated that the tension per cross-sectional area unit is greater for type 2 or fast twitch fibers than for type 1 or slow twitch fibers. So myofibers actually receive blood flow from surrounding capillaries and are commanded to contract and relax by what's known as the neuromuscular junction. The connection between our brains and muscles is an important consideration. The neuromuscular junction, or the NMJ, refers to the area between a motor neuron and the skeletal muscle fibers. Acting as a bridge, electrical signals from the nervous system are translated into muscle contractions. This synapse occurs at a specialized area of the sarcolemma called the end plate. So at the NMJ, the distal end of each branch of the motor neurons will release neurotransmitters such as acetylcholine, which binds to receptors on the muscle fiber, triggering an electrical impulse that leads to the muscle contractions and the mechanism that we just discussed beforehand. Looking into NMJ physiology, a motor unit consists of one motor neuron in the spinal cord and the muscle fiber it innervates. Motor units per muscle range from 100 for a small hand muscle to 1,000 or more for large limb muscles. They have also been shown to differ greatly in the force generating capacity. Involuntary contractions, force is modulated by a combination of motor unit recruitment and the changes in motor unit activation frequency. The greater the number of motor units recruited and their discharge frequency, the greater the force will be. Connecting back to what we just learned about fast and slow twitch fibers, it has been found that the NMJ of fast and slow twitch muscle fibers differ in their transmitter output and therefore affect the fatigability of these fibers. Nerve terminals that supply to fast twitch, or those type 2 fibers which we talked about, have been shown to present greater synaptic surface area and contain more vesicles than those supplying slow twitch fibers. There is also accumulating evidence that the NMJ in mature skeletal muscles are not fixed permanent structures, but instead are continually remodeling and therefore possess a large degree of functional plasticity. The most dramatic form of this is when the NMJ becomes fragmented and ultimately dissolves following nerve transection, what's known as denervation, concurrent with axon withdrawal. If the surrounding perisynaptic Schwann cell, which act as support cells along the neurons, are ablated, it will also result in rapid axonal retraction and NMJ destabilization, both structurally and functionally. These have been found to play a crucial role in muscle remodeling, with increased integration at the NMJ during development and their intimate contact with perisynaptic stem cells known as satellite cells. We will have a video covering muscle regeneration and satellite cells, so make sure to check that out if you want to learn more about how muscles are generated in the first place. In this case, it's important to understand that NMJ remodeling ultimately affects neuromuscular transmission. Now, normally, neuromuscular transmissions are highly reliable, as each nerve impulse results in the release of more neurotransmitter than that is required for evoking an action potential in the muscle fiber. This release of surplus transmitter and consequent axis depolarization of the postsynaptic action potential via neurotransmitter receptors is often referred to as the safety factor, which ensures that a postsynaptic action potential will occur in response to each nerve impulse, at least in healthy tissues. We will look into how neuromuscular junction remodeling can impair the reliability and efficiency of neuromuscular transmission in a future video, so make sure to check that out. So I guess to summarize, the purpose of today's episode was to lay the building blocks for future application of muscle weakness and muscle conditions, particularly sarcopenia. This episode delved into the fascinating world of muscle strength, exploring its connection to the microscopic building blocks of muscle and the communication system that makes it all work. We've learned that muscle strength isn't just about size, but also about the myofibril, the tiny contractile unit within the muscle fibers. Myofibrils are packed with even smaller filaments called actin and myosin. These filaments slide past each other during contraction, generating the force that allows us to move. We then introduced the neuromuscular junction, the critical link between our nervous system and muscle fibers. The NMJ acts like a message relay center, translating nerve signals into muscle connections. By understanding these connections, we can appreciate the complex machinery that underlies even the simplest movement. So that's all we have for today on Kinesthetic Knowledge. I'm your host, Isha, and I will see you next time. Take care.